All right, now we're going to talk about glycoengineering. So, what if I told you that one of the biggest obstacles preventing plants from making human medicines wasn't the protein itself? It's the sugar. And I'm not talking about table sugar. I'm talking about tiny sugar molecules that are attached to proteins that determine whether a drug actually works inside of the body. So, when we think about biological drugs like monoclonal antibodies, we usually focus on the protein, but proteins aren't the whole story. After they're made, cells attach complex carbohydrate chains called glycans. This process is called glycosylation. Think of glycans like the shipping label and barcode on a package. The protein is the package. The glycans tell the immune system where it belongs, how long it should stay in the bloodstream, and sometimes whether it works at all. For over 40 years, Chinese hamster ovary or CHO cells have dominated biologic manufacturing because they naturally produce glycans that are very similar to humans. That's one reason regulators trust them. But CHO cells are expensive and they grow slowly, and manufacturing facilities cost hundreds of millions of dollars to build. So why not use plants? Plants can manufacture proteins incredibly efficiently. They're inexpensive. They scale beautifully. They're much harder to contaminate with mammalian viruses. There was just one major problem. Plants don't decorate proteins the same way that humans do. A plant naturally adds sugars that humans rarely produce. So early researchers worried that these plant sugars might trigger unwanted immune reactions or alter how the biologic drug actually functions in the body. So enter glycoengineering. This is where the science becomes fascinating. Instead of accepting plant glycosylation, scientists rewired the plants. Using genetic engineering, they turned off the enzymes responsible for adding plant specific sugars. Then they inserted human enzymes that build human like glycans. The plant is still making the protein, but now it's decorating that protein with sugars that closely resemble those produced in human cells. This changes almost everything because glycosylation influences drug stability and half life and immune recognition and antibody activity and safety. And in some cases, researchers can even design glycans that improve antibody performance beyond what's typically produced in traditional CHO cells. Here's where plant systems become especially interesting. CHO cells naturally produce a mixture of glycan patterns. Plants can often be engineered to produce a much uniform glycosylation profile. That means every antibody coming off the production line can be more consistent, and consistency is exactly what pharmaceutical companies want. As a veterinarian, I find this especially exciting. If plant based biologics lower manufacturing costs, imagine they could make advanced therapies like monoclonal antibodies and growth factors and cytokines and vaccines and regenerative proteins way more accessible for veterinary patients. Many biologics are currently limited not because we don't know how to make them, but because they're too expensive to manufacture. Glycoengineering has transformed plants from simple protein factories into highly sophisticated biologic manufacturing platforms. The question is no longer, can plants make biologics? They clearly can. The real question is, can they manufacture them faster, more consistently, and more affordably than CHO cells? The answer may reshape the future of medicine. So if you've been following this series on plant-made biologics, let me know what topic you'd like me to cover next. Should we talk about transient expression, molecular farming, downstream purification, or maybe how companies like phytoceuticals are using these technologies to transform biological manufacturing?